What's up, everybody? Herman here again. Today we're going to be talking about population genetics. So let's jump right into it. So we're going to be talking about the genetic structure of populations. We're going to talk about the Hardy-Weinberg law and how to calculate it, genetic variation in space and time, and then forces that change gene frequencies. So genetic structure of populations. It refers to the distribution of genetic variation within that population. These are some key aspects of the genetic structure of populations. Genetic variation, the presence of different alleles of a gene within a population, allele frequency, the proportion of a particular allele with a population within a population, genotype frequency, the proportion of individuals with a specific genotype or a pair of alleles within a population. We have a gene pool, the total genetic information contained within a population. Some factors that influence genetic structure, population size. Small populations tend to have less genetic variation than larger populations. Mutation rates, the rate at which new mutations occur within a population. Gene flow, the movement of individuals with different genes into a population, which can increase genetic variation. Genetic drift, random events that can alter the frequency of alleles within a population. Selection, natural selection can favor certain alleles over others, altering the genetic structure of a population. Types of genetic structure, mixia, a population with random mating and no genetic structure. Subpopulation, a population divided into smaller subgroups with distinct genetic structures. Metapopulation, a population composed of multiple subpopulations connected by gene flow. Understanding the genetic structure of populations is essential in fields like evolutionary biology, conservation genetics, and epidemiology. The Hardy-Weinberg law. This is a fundamental principle in population genetics. It describes how genetic variation is maintained in a population over time. The law states that allele and genotype frequencies will remain constant from one generation to the next, assuming certain conditions are met. Random mating, individuals mate randomly with respect to genotype. No mutation, no new mutations occur, no gene flow, no individuals enter or leave the population, no selection, all genotypes have equal fitness. Large population size, the population is sufficiently large. Calculating the Hardy-Weinberg equilibrium. To calculate the expected genotype frequencies under Hardy-Weinberg equilibrium, you can use the following equations. So let P, which is equal to frequency of allele A, Q equals the frequency of allele A, P to the power of 2 equals frequency of genotype AA, capital AA, and P equals the frequency of allele capital A. So P to the power 2 is the frequency of genotype capital A. 2PQ equals the frequency of genotype A, capital A, lowercase a. Q to the power of 2 equals the frequency of genotype lowercase a. So the Hardy equation is P to the power of 2 plus 2PQ plus Q to the power of 2 equals 1, where P to the power of 2 represents the frequency of the homozygous genotype AA. 2PQ represents the frequency of the heterozygous genotype capital A, lowercase a, 2PQ represents that the genotype capital A, lowercase a. Q to the power 2 represents the frequency of the homozygous genotype lowercase aa. So suppose we have a population with two alleles, A, capital A, and lowercase a. The frequency of allele A is 0.6. The P equals 0.6. We said capital A, right? P equals capital A. And the frequency of allele A, lowercase a, is 0.4. So Q equals 0.4. Using the Hardy-Weinberg equation, we can calculate the expected genotypes frequencies. And so P to the power of 2 equals 0.6 to the power of 2, which equals 0.36. So that's the frequency of genotype capital AA. 2PQ equals 2 times 0.6 times 0.4, which equals 0.48. So the frequency of genotype capital A, lowercase a, Q to the power of 2 equals 0.4 to the power of 2, which equals 0.16. The frequency of genotype lowercase aa. Therefore, the expected genotype frequencies under Hardy-Weinberg equilibrium are capital AA 36%, capital A lowercase a 48%, and lowercase aa 16%. Genetic variation in space and time. So genetic variation in space and time refers to the study of how genetic differences are distributed across different geographic locations and how they change over time. Types of genetic variation, spatial variation, genetic differences between populations or individuals at different geographic locations. I think a simple one would probably be like skin color. People that tend to live closer to the equator tend to have darker skin tones, and that's just because the rays of the sun are stronger there. So the higher melanin absorbs more of the ultraviolet rays. I guess that's just one example we can mention real quick. Temporal variation, genetic changes within a population over time. Some factors influencing genetic variation, mutations, of course. New genetic variants arise through mutations, gene flow, movement of individuals with different genes into a population, genetic drift, random events alter the frequency of genetic variants, natural selection, environmental pressures favor certain genetic variants, genetic hitchhiking, neutral or deleterious alleles become more common due to linkage with beneficial alleles. Methods for studying genetic variation, phylogeography, analyzing genetic data to reconstruct evolutionary relationships and geographic distribution within and between populations. Oh, sorry. Genome-wide association, GWAS, we've mentioned this before, identifying genetic variants associated with specific traits or diseases. Examples, human genetic variation, studying genetic differences between populations from different parts of the world, species divergence, analyzing genetic changes that occur as a species diverges and adapts to new environments. Adaptation to climate change, investigating genetic changes that help populations adapt to changing environmental conditions. Understanding genetic variation in space and time helps us reconstruct evolutionary histories. So this is rather important. Identify genetic factors underlying adaptation. Identify genetic factors that underlie adaptation and disease. So connecting histories in order to find out why certain diseases happen. Inform conservation efforts and management of natural populations. Humans are going extinct, right? Or even animals, you know, we can apply that, or basic life, right? And develop personalized medicine and targeted therapies. So gene therapy, of course, probably the biggest one. Forces that change gene frequencies. So there are different types of forces and or variances. Forces that change gene frequencies, also known as evolutionary forces, are the mechanisms that alter the frequency of alleles or the different forms of a gene within a population over time. So these forces can act alone or in a combination to shape the genetic makeup of a population. The process by which new alleles are created through errors during DNA replication or repair, mutations. And they can be beneficial, neutral, or deleterious. They can cause issues. Gene flow, the movement of individuals with different genes into a population, which can increase genetic variation and change gene frequencies. Random events such as genetic sampling, errors, or natural disasters that can alter the frequency of alleles within a population. This is known as genetic drift, and it can lead to the loss or fixation of alleles. The process by which environmental pressures favor the survival and reproduction of individuals with certain alleles, leading to an increase in their frequency within the population. And that's natural selection, of course. Sorry. Genetic hitchhiking, the process by which neutral or deleterious alleles become more common because they are linked to beneficial alleles that are under positive selection. Artificial selection, the intentional selection of individuals with desired traits by humans, which can lead to change in gene frequencies within a population. That is kind of crazy. Genetic draft, the effect of genetic drift on linked alleles, which can lead to the hitchhiking or neutral or deleterious alleles. These forces can act on different scales from small populations to entire species and can influence the evolution of populations over time. So that's all I got for you guys today. I hope you guys learned something. It's always a pleasure to share this information with you guys. Yeah, I mean, if you guys have any questions, you could always leave a comment, or you could use the internet. Internet is a great tool. But yeah, I'll see you guys in the next video. Peace.